how to treat these disorders, and I'm sure we are going to have a very illuminating talk from Dr. Mohankumar regarding gene editing to prevent these disorders.
Thank you, Chairperson. So, like you have heard the talk about past 3 talks about various variants that are associated with pancreatic disorders. So now my work is grossly talk about how we can use the new tool like the CRISPR-Cas9, and there's a traditional version and also the advanced version. How we can use these tools to correct these variants so that we can reverse the disorders. So most of my work is based on hematologic disorders because we can take the cells out and then we can manipulate. So I will show the proof of principle concept in the hematologic disorders, which can be also applied for the various other disorders including pancreatic. So the overview of my talk, I'm going to just introduce about the CRISPR-Cas9 and its mechanism and variants, and then how it can be applied, and whether we can do it by ex vivo and in vivo platform, and then also about the current work going on in my lab. So coming to genome editing, so it can be applied to both genetic disorders and also for non-genetic, because if you look at the variants that are present in various disorders, if you look at in the bone marrow, because there are various disorders caused by pathogenic variants that causes lysosomal storage disorders like sickle cell anemia and beta thalassemia. And then if you look at liver, there are hemophilia, and then muscle muscular dystrophy in lung and cystic fibrosis. So there are a variety of monogenic disorders can be corrected using this platform, and some of them including non-monogenic disorders like cancer and infectious disorders like HIV and leukemia, and also can be corrected by using the CRISPR-Cas9 platform. So everything started with double-strand break, because if you want to correct any pathogenic variant, you have to make a double-strand break at the target locus. So this was first done in 1994 by Maria Jason group, and they made a double-strand break using replicating enzyme, but it's very infrequent. And later there a lot of development happened to make a double-strand break, but particular locus with high frequency. So started with zinc finger nuclease, and then later with FOC1, and then later with Cas9. But problem with ZFN and then with Talensar, is the protein engineering, because if we want to target one particular locus, we have to do a lot of protein engineering to target the particular locus. But for CRISPR-Cas9, by just changing the 20 base pair, the guide RNA sequence, we can target any part of the genome. So basically it started with the bacterial immune system. The CRISPR-Cas9 has a defense platform identified from the bacteria, to prevent from the bacteriophage infection. So they identified the Cas9 from this, and then later they used for the genome editing purpose. They repurposed for editing the particular part of the genome. So once we have the Cas9, it makes a double-strand break, and after that we can make 2 things happen. One is we can use the NHEJ pathway, and other one is the HTR pathway. In case of NHEJ pathway, we can disturb the gene, like the pathogenic variants that are causing the disease, we can disturb by using NHEJ pathway. Or in other case, we can correct the pathogenic region using by donor template. It's called HTR mediated repair. So advantage of CRISPR-Cas9 system is very simple, and it's cheaper, and it shows better efficiency compared to all other platform. And then we can target multiple sites simultaneously using this platform. But the problem with CRISPR-Cas9 is because it's making double-strand break, it can lead to p53 activation, and can also causes apoptosis, and then it can also lead to larger deletions, because we are not going to make a deletion only in the target genome. It can also make a deletion in other part of the targets, which can lead to larger deletions. To overcome this, recently there are a lot of new platforms has come up. One is called base editing, another one is prime editing. Both use single strand break mechanism to make a base pair conversions. So if you look at base editing and then prime editing, the base editing converts one base pair to other base pair, and without making a double strand break, because basically it deaminates one nucleotide to other nucleotide without causing any DNA break. Similar thing if you look at prime editor, there are limitations with the base editor because it can only convert transitions, but if you want to convert the transversions, it could be possible only with prime editor. So the whole thing is now we have 3 different platforms for using Cas9. One is by using the double strand break mediated, either by knock-in or knockout. And by single strand break, we can use it by base editing and prime editing to correct the disorders, or by using dead Cas9 based approach, where we don't even make single strand break, but we can activate and then repress the genes. And all this platform can be done by ex vivo or by in vivo. But in case of like hematology disorders, this is advantage, because if you want to give all the Cas9 platform to treat in different solid malignancies or in different diseases, like we have to give it the platform in vivo. That time we get a lot of immune response. To avoid that, like we can take out the cells ex vivo, like in sickle cell and beta thalassemia, we can take out the stem cells from the bone marrow, and then we can correct the corresponding disease causing mutation by using this platform. And in addition to that, like but for the dead Cas9 platform, like it doesn't cause us any double strand break, but we can activate and repress the genes. But if you look at, if you want to make any modeling of the disease, or if you want to cause a particular gene suppression, we can use this model to activate and repress accordingly. And these are various platforms, like the gene editing is always used for, like a infectious disease, like in case of here, like HIV1, they prevent the CCR5 receptor by making a knockout, so that HIV cannot enter, and then we can prevent the disease. And similarly, in case of hemophilia, like we can make the inversion correction, because that's happening in more than 50% of the population in hemophilia. So we can correct even the large inversion using CRISPR-Cas9 based platform. And one of the main contribution is like the exon skipping. If you look at muscular dystrophy, most of the things are happened by exon skipping, and we use CRISPR-Cas9 platform to like causes the disease causing the exon to overwrite, so then we can have the normal gene expression. Similar thing in the cancers. Not only we can target one genes, we can target multiple genes parallelly using this system. So by using two different guide RNAs targeting on the two different locus, we can overcome the problem. And also we can activate and suppress the genes. In case of if we want to make iPSCs from the particular cell type, we can use this platform to make the gene activation accordingly. And like laterally, like these technologies are always used in correction of sickle cell model. Like the sickle cell disease was corrected using the HTL based approach using Cas9, and also like the viscous autism syndrome, also like we can put the whole gene in particular locus by using the CRISPR-Cas9 system. And like also if you want to stop any gene expression, we can use a base editor, because it can convert one base pair to other base pair. We can stop the gene expression by using base editor with higher frequency. So, and this also can be used for exon skipping in the DMT. And in our lab, we used for sickle cell correction using the prime editor, because we want to use the double strand break-free approaches. So we took the human erythroid cells, and then we corrected the sickle mutation by using prime editor with higher frequency. And then after correction, we can see that like the RPC is not sickling, and then it shows normal phenotype. So coming to pancreas, like this platform can also be used in pancreas, but one of the problem is we have to do it in vivo. But there are few advantages, like if you want to model any pathogenic variants, like either from GWAS studies or from any pathogenic variants, we can model those variants to see whether it can cause the disease phenotype. So we can model those mutations by using particular cell line and using CRISPR-Cas9 platform. And we also, like instead of doing the whole genome screening, we can also do the screening with the CRISPR-Cas9 to identify the variants that are associated with the disease progression. So by doing the screening, like with all the multiple targets, we can identify what are the mutants that are associated with the particular disease progression. And in addition to that, like the CRISPR-Cas9 platform is also used for identifying the variants that are associated with the pancreatic cancers. Again, as I mentioned before, like we can do the whole screening to identify the mutants that are associated with the cancer progressions. And later we can correlate this with the patient sample and then see whether these are the causative factor for tumor progression. And in our lab, like we use the CRISPR-Cas9 tools, like mainly, coming to our work, like we use this for elevating the fetal hemoglobin for treatment of sickle cell and beta thalassemia. So now we are taking this work for the phase 1 clinical trial at the moment. And we also use both the base editor and also the prime editor for correcting the sickle cell disease, and also for creating the model for beta thalassemia, and also for correcting the beta thalassemia with higher efficiencies. And in addition to those, like we also work on LN, severe congenital neutropenia, so where is also caused by mutation in the elastase gene. So we try to overcome this by doing exon skipping. And now we also created a new base editor tool which overcomes the deletions. So we made a Nicky's division base editor which does not make any double strand break, but it converts one nucleotide to other nucleotide with higher frequency. And this also we are using it to make it a safer approach for screening the genes. And we are taking this work forward, and we are using this for the clinical trial for treating the sickle cell disease and beta thalassemia at the moment. And this is my group, and most of my group is working currently in the hematologic disorders, and funding is from India Alliance and DBT and DST for the support. Thank you.
Thank you, Dr. Mohankumar, for introducing us to the basics of CRISPR-Cas9 based gene editing. One naive question to you. I mean, if you were to do some gene editing in vitro, how would you target the pancreas?
Yeah, so basically we need the basic thing, or the Cas9 based platform has two components. One is the guide RNA, another one is the common nucleus domain. So we can target the nucleotide sequence wherever we want to target. By doing that, we can target any part of the gene, as long as it has a PAM sequence. But recently there are a lot of new variants has come up which use non-PAM variant also. So basically we can target any part of the genome by using CRISPR-Cas9 platform.
Delivery, I mean delivery into the target organ.
So delivery, if you want target, like,
the pancreas, for example. For example, if you were to edit a gene causing, say, pancreatic cancer or some chronic pancreatitis gene.
So basically, there are viral vectors are mainly used, and then non-viral.
Same standard viral.
Yeah. So like AAV virus and antiviral, and we also have LNPs like lipid-like nanoparticles, and we can use both to make it to target to a particular part.
So once you are using vectors, then won't they die out? I mean, for example, the viral vectors, if you use, would this the delivery of this edited genes have to be repeated? Because unless you give it to dividing cells, it's not going to populate the organ. So would you need to keep giving repeatedly the gene therapy, and how expensive would that be?
So coming to everything we are doing in ex vivo, but like I can correlate that with in vivo also. But if you want to do it in anything of the pancreatic related disorders, we can do it one time, and then we want to be transient, because these are the protein that's not expressed normally in the body, because the Cas9 is derived from the bacteria. So if you express for a longer time, that may give us to immunogenicity, that also can eliminate the cells. To avoid that, we want to be transient as possible. So for that, we use the AAV vector. The AAV vector makes the delivery very transient, unlike a lentiviral vector. So by doing that, we can overcome the immunogenicity.
So AAV vector, will it get integrated? Even you need something to integrate into the genome to be able to have a long-lasting effect. Because AAV vector is there, and when that cell dies, the effect dies.
No, ma'am. We were talking about making mutation and correction by using the...
Maybe something wrong in my understanding.
So we want to use this platform to make a genetic correction transiently. Because if you give the base editor or Cas9 protein, it makes changes in the genome, but we need the protein only for transient time, like within 12 hours we want to express. Okay. After making 12 hours, the mutation is made in the cells, so it won't get eliminated.
Thank you so much.